Everyone, let me know if something goes wrong, if you can't hear me or see the screen properly. Yeah, I'm Jillian Petersen. I'm coming to you from the University of Vienna, and I work at a fantastic center called the Center for Microbiology and Environmental System Science. We're about 150 people. Most of us work on microbial ecology. So scientifically, it's a wonderful place to work as a microbial ecologist. We work on topics from the deep sea to shallow water, sediments, water column, the human gut, soils, you name it, it's being researched in this department. So it's really a stimulating environment. I'm going to talk today about nitrogen fixation and chemosynthetic symbionts, two topics I hear that you've been introduced to before, but I have also heard that if you get lost, you'll stop me, so that's great to hear. And I hope that I can tell you a little bit more in depth a story about a current research project on these topics. And I've termed this talk from ecosystem functions to evolutionary context. As Chloe already mentioned, the topic of research in my lab is about animal microbe associations. And I just wanted to briefly introduce the amazing diversity of things that microbes can do for their animal hosts. And this, of course, is the basis for these advantages that animals gain from microbes is the incredible metabolic diversity that microbes have, that animals just don't. And so the only way of tapping into these functions is to establish beneficial associations with microbes. And this has happened again and again and again over evolution. There is barely an animal or plant alive today that does not rely on microbes for some kind of beneficial function. So to start off with, they can break down complex polymers. This happens every day in our own guts and in the guts of all animals that have guts. They can synthesize things that are missing from an animal's diet. So an example of this would be sap-feeding insects that synthesize amino acids when the insect doesn't get these from the plant diet. This may also happen in our guts with the synthesis of vitamins by gut bacteria. They can synthesize toxic substances that may protect the host against predation. And this happens in bivalves, in sponges, in insects, in many different animal types, but very common in those that have a lifestyle where they're stuck on a hard surface and can't move, can't leave if they're being predated on. That's a defensive type of symbiosis. They can also destroy toxic substances. For example, in insects, there are a lot of microbes that associate with the host and destroy the toxins that they take in through their plant diet. So kind of the other way around. They can make light from food. A very well-known example of this is the Vibrio bacteria that associate with marine, with a squid. They can also make food from light. And this is the example of corals, where the symbionts use light energy to synthesize CO2, and they pass on this as a nutrient source for their host. But there are also bacteria, and these are the ones I'm going to talk about today, that make food from reduced chemicals. So in the same way, and these are called chemosynthetic symbioses. So in the same way that in a photosynthetic symbiosis, the host does primary production using the energy it gains from sunlight. In chemosynthetic symbioses, here's an example from the deep sea in a mussel called Bathymedailus. If you zoom in and you could look into the tissues of these animals, you'd see that they're packed with chemosynthetic bacteria. And these bacteria are also primary producers, but instead of using sunlight, they're using chemical energy to power this process. But they do provide a source of nutrition for their animal hosts. In the deep sea, this is very important because there is no sunlight to power the ecosystems of animals found in the deep sea. So the evolutionary innovation here was that animals farm these primary producers within their own bodies instead of filtering them out or eating them from the environment. So although I worked on deep sea symbioses, I've worked on this animal here, Bathymedailus mussels, for many years. My lab has now set up shop working on a chemosynthetic symbiosis from shallow water ecosystems. I'm often asked, Vienna doesn't Austria doesn't have a coastline. Why do marine research in Austria? Well, sure, I love the marine environment. The health of the oceans is relevant to us all. But it's also, I'm also much closer here to the Mediterranean. This is the Mediterranean where we do most of our field work than I was in my lab previously at a marine research institute. And we're looking at really fundamental questions of interactions between hosts and microbes. So that's my answer to that. The family of clams that we've chosen to work with are called lucinids, and they have a partnership, a chemosynthetic symbiosis with sulfur-oxidizing symbionts. They're often called moon clams, at least in German, they're called moon clams because of their typical shape and luminescent color. And you can sample them via scuba diving. No need to have this kind of deep sea infrastructure and big research vessels. We can just go out with buckets and our hands and dig through sediments surrounding seagrass meadows, and we often find, this is where we often find lucinids. Like the deep sea chemosynthetic symbioses, they host their symbionts inside of a specialized organ, the gill. This is the filter feeding organ and also the gas exchange organ, even of bivalves that don't have symbionts. In these symbiotic bivalves, it's really been repurposed as a home for the symbiotic microbes. If you look at them in a cross-section through some of these gill filaments, you can see the symbiotic bacteria stained in a purple color. The blue shows the host tissue. These are the nuclei of the host that have been stained with a specific DNA stain. And if you zoom in a little bit closer, you can see that these are intracellular symbionts that really pack the gill cells, the epithelial cells of the animal. And so, as I said, it's been repurposed as a home for this massive population of intracellular symbionts. So going back to their environment, they're often found in seagrass sediments. And just like coral reefs, seagrasses need sunlight for growth. They need these very nutrient-poor conditions so pristine oligotrophic conditions. And so just like coral reefs, these are typically considered as nutrient-poor habitats, particularly in nitrogen. A few years ago, we were out in Elba sampling the clams and looking at their environment. And a postdoc who was in the lab, Ulisse Cardini, who now runs a research group in Naples at the Marine Station. He did some measurements of nitrogen, available nitrogen, dissolved inorganic nitrogen and phosphorus in the habitat of the clams. So this is just sampling through, 0 would be the top of the sediment, and then taking samples throughout the depth of the sediment. And this gray box here is about where you would find the clams. They live buried inside sediments. And so in the habitat of the clams, we do have a fairly nitrogen limited environment with inorganic nitrogen down in the single digit micromolar range. This changes a little bit through the seasons, but it's the nitrogen limitation typically goes throughout the entire year. And so a couple of years ago, we were looking into these clams. We'd spent many years studying symbioses in the deep sea, but nobody had really been looking into these clams in any detail. So we started looking with genomics to start with, sequencing the entire genome of the symbionts to find out what they could do, what they were capable of. And unexpectedly, we found a NIF gene cluster, which is the cluster that's responsible for biological nitrogen fixation. And this was surprising because at the time that we sequenced this genome, there were many genomes available for chemosynthetic symbionts from all over the world in many different habitats, and none of them had been shown to be capable of fixing nitrogen. They were all capable of fixing carbon, but this was the first one that was also able to fix nitrogen. It's not enough to only have the genes in your genome. They also have to be expressed to be functional. And we did transcriptomic, metatranscriptomic experiments, looking at all of the genes that were expressed by the symbionts inside the host, and also the proteomes to look at proteins that we could detect inside the host, and found these NIF genes expressed at a very high level by the symbionts in the host. So indicating that this nitrogen fixation capability was probably active in the symbiosis. I won't go into too much detail about the isotope data, but we looked at the natural abundance of stable nitrogen isotopes, and this was also consistent with active nitrogen fixation, providing a source of nitrogen to the host. Happy to go into that in more detail later. We also found, I didn't mention, this is a very diverse family of bivalves. There are probably 500 species of leucinid clams in our oceans today, alive in our oceans today. There are many more fossilized species that we know very little about. And we looked at many different species, the symbionts of many different species from around the world, and found that this NIF-H gene was very common in leucinid symbionts. And so nitrogen fixation seems to be quite widespread in leucinid symbioses. So why were we so interested in this? Usually a village of diverse symbionts supports animal health. This is most animals we know, including the example of corals. They rely on a community of microbial associates like we do, with incredibly diverse microbes found in our guts, each of them fulfilling one specific role in the symbiosis, providing one function to the host. In the example of corals, you've got the photosynthetic symbionts that provide carbon, and you've got other symbionts such as bacteria that provide nitrogen. So this is partitioned into different members of the symbiotic community. And the leucinids are interesting because this is they've evolved a different strategy. They've got this very specific symbiosis with one symbiont type that's an all-rounder. It can fix carbon and it can fix nitrogen. And if you contrast this with other very well-known nitrogen-fixing symbioses, I could have chosen many from the marine environment, but this is just an example from terrestrial plants. In the plant model, you've got the photosynthetic partner that's providing carbon to the nitrogen-fixing partner, which then provides nitrogen. So it's an exchange of carbon and nitrogen. And this is a contrast to the leucinid clams, where the symbionts seem to be providing both. So this was published quite a few years ago now. And since then, we're starting to realize that there may also be an ecological role for nitrogen fixation by the chemosynthetic symbionts of leucinids. And there are many studies now showing also that the seagrasses benefit when they associate with leucinid clams. So you can keep seagrasses in mesocosms in the lab with or without these bivalves. And the seagrasses tend to grow better when they're cultivated together with the bivalves. So there may also be some mutualism, a three-way mutualism between seagrasses, the bivalves, and the symbiotic bacteria of the bivalves. And it is accepted that one of the benefits of associating with leucinids is that their sulfur oxidizing symbionts remove this toxic sulfide from the sediments. And this allows the plants to grow better. But there's also a theory, now that we know they can fix nitrogen, that they might actually be providing a source of nitrogen in the seagrass habitat. But that hasn't been shown experimentally yet. This being a typically nitrogen-limited habitat, this may play a role. Other ecological roles are known for leucinid clams. There's one really cool example from a Caribbean island where lobsters, spiny lobsters that are fished, they're a very important economic resource in these islands. They gain a lot of their nutrition from leucinids that they predate upon in seagrass sediments surrounding the islands. And so there are places on Earth where chemosynthetic production really also provides an economic benefit. I mentioned the ecosystem role of leucinids in seagrasses, but so what if it's not based on hydrogen sulfide removal, but on nitrogen provision? This would be a new aspect in this mutualism between seagrasses, bivalves, and bacteria. And we wanted to look at this a little bit experimentally. So we had this hypothesis that the symbionts of the leucinid clams providing nitrogen to the environment. How could we show that? And the hypothesis was that, that they would be releasing a source of nitrogen to the surrounding sediments. And to measure this, we used some isotope tracers. We used 15N2, the rare isotope of nitrogen, in incubations with leucinid clams. We also added a carbon isotope label, inorganic carbon, that was enriched in the rare isotope carbon-13. And this can be measured over time with mass spectrometry. If you incubate your animal with these isotopic tracers over time, you can see that they become enriched in the tissues of the animal. And so to measure active nitrogen fixation and carbon fixation by the bivalves, we incubated these in 2 different seasons. And what we saw was we could measure active nitrogen fixation in the bivalves with this method in 2 seasons, in both spring and fall. And we could also measure carbon fixation with this method as the tissues became enriched in the rare isotope as measured by mass spectrometry. So this was an indication for a start, the first experimental indication, first experimental evidence that the symbionts were actively fixing nitrogen. The next question is, was some of this being released to the environment as some kind of useful nitrogen source for the surrounding environment, such as the seagrasses? To do this, we used a technique that's been used very often in terrestrial science, but not so often in the marine environment yet. And I'm happy to go into this in more detail later, but just to give you a very superficial idea of the experiment, it's called the technique is called isotope pool dilution. And so the problem with using these using isotopes in experiments is that if you've got a biological process, it could be consuming and producing your compound of interest in the incubation at the time. And so if you measure in the beginning and at the end, that's only a summary of consumption and production in the vial over time. And if you want to tease these 2 apart, what you can do is have a pool of the compound of interest that is a pool that's separated into 2 different fractions, one that's labeled with the rare isotope and one that contains mostly the common isotope, 14N in this case, in this example. And there are many assumptions underlying this method. I won't go into them now, but in general, if you've got a productive process, only one of these will be produced, the 14N, because your natural system has mostly 14N. So if in the bivalve, for example, any kind of ammonium is produced and secreted to the environment, that will be 14 ammonium. So if you've labeled your ammonium pool in the beginning with some 15N and the bivalve is producing 14N, if you know the fraction of each of these and the total concentration, you can then work out, you can tease apart consumption versus production. Only 14N will be produced, but both, if they're being consumed, both will be consumed from the pool in the incubation vial. A little bit tricky, but I'm happy to talk about that more later. I think the main message that you should take from this is that it's possible then to come up with rates that are net consumption or production in incubations with these isotopes. And so in a bivalve that's got symbionts that might be using ammonium, but also producing it and releasing it, we were able to, with these experiments, show that there was a net release of ammonium to the environment if we incubated the bivalves in a vial with some artificial sediment. And so we could quantify how much ammonium would be released if these rates are really representative of what's happening in the environment, and show that there is large potential for ammonium production and release by leucinoid bivalves to the surrounding environment. And it's probably based on the nitrogen fixation capability of their symbionts. So I've talked up until now about one type of environment. Sorry. But throughout their long evolutionary history together, leucinoids, we have fossils from 400 million years ago. So this is a very ancient symbiosis. And we also have some indication that even these ancient fossils from the Silurian had a chemosynthetic symbiosis. So this is really one of the oldest partnerships known. And throughout this long evolutionary history, they have together conquered many different habitats, including these reef lagoons, these seagrass meadows, but they've also been found at hydrocarbon seeps and mud volcanoes on the deep sea, extreme environments in the deep sea, hydrothermal vents, in mangrove swamps, rocky rubble, very different types of marine environments. And we also had the question, given that the nitrogen availability really differs a lot in these different habitats, is nitrogen fixation conserved in all leucinoid symbionts? And at the time we started out, there was very little known about the biodiversity of the symbionts of leucinoid clams. There was, strangely enough, a lot more known about the biodiversity of the hosts, because there's been work done on that, taxonomic work over many decades. A researcher called John Taylor at the Natural History Museum in London has been working on describing these species for most of his career. And so this is why we know there are probably 500 or at least 500 different species of hosts in the oceans today. But on the symbiont side, we know very little. And so when we started out this research, there were a couple of genomic studies. Here was our work from Elba in the Mediterranean, and there were a couple of studies on looking at the genomic diversity of symbionts in the Caribbean. But very few studies and on the symbiont diversity. And most of these were on, obviously on bivalves from shallow waters, because these are much easier to sample. Most of the known species, most of the species we know are from shallow water sites, but this might be a sampling bias. Most of what we've explored is in shallow water that's easy to access. But a few years ago, an expedition set out from the Philippines that aimed to look at a depth gradient, look at biodiversity of animals in sediments, moving away from islands, the coastal regions of islands in the Philippines, and sampling down into deeper depths. And John Taylor, as the leucinoid expert, was on this expedition, and he looked at the diversity of the clams that he found across a depth gradient in all of these samples. And surprisingly, he found that although the deepest samples tended to have a lower species diversity, they still had a remarkable number of species in each sample. And the highest diversity was even found below a scuba diving depth. So 0 to 70 would be the most intensively sampled depth. And below that, there was still a massive diversity of leucinoid bivalves that have been really understudied and undersampled. So they're typically considered a shallow water symbiosis, and yet this is probably just because we haven't looked hard enough and haven't really characterized the diversity at depth. But we do have a few samples, and that's what I'm going to talk about today. If we look at now the question, going back to the question of nitrogen fixation and whether this is conserved in all of the leucinoid symbionts we look at, thinking about the environments that we're considering here. The coral reefs and seagrass sediments, as I mentioned, are typically nitrogen limited. But as you go deeper waters are not typically nitrogen limited. So there's no phototrophic primary production, and so inorganic nitrogen doesn't get used up quite as much as in shallow waters. That's a terrible generalization, but it's more or less a pattern that's seen commonly in the oceans. And measurements showed that at cold seeps, for example, and hydrothermal vents, we've got 10 times as much available nitrate as we do as we saw on Elba, as we have in shallow water sediments typically. And ammonium can also be very abundant. And so here's an environment inhabited by lucynid clams that is actually not like nitrogen limited. There's plenty of nitrogen available. And as I mentioned, we do have some samples from deep sea lucinids through many years. I think the 6 years that I've been in Vienna, we've had through a network of collaborators, we've tried to get our hands on every lucinid sample we possibly could. And this has really been led by a postdoc, Benedict Yuen, and a PhD student, Jay Osbartik, in the lab, who put a lot of effort into searching natural history museum catalogues or museum catalogues, and also going out into the field themselves to try and fill out our sample set to really cover the entire diversity of the host animals, and also to try and get samples from as many different environments as possible. So as I mentioned, this also involved collaborations with museums. This is John Taylor. We have some samples that were collected 100 years ago that we were able to play with in our labs, and we've gotten genome sequences from symbionts out of these incredibly valuable samples. But we've also been out in the field collecting in the days before COVID. This was an intense effort. But now we do have a sample set and a data set that really covers the diversity of host animals. These are all so these are the locations that we have samples from. These are the host genera. There are, we've covered 53 host genera so far. I'm not sure how many host genera have been described, but this is really representative of the whole diversity of the lucynid tree of life. We covered 85 different species of hosts from deep and shallow waters. And from each of these host animals, we've been able to take a sample and through metagenomics sequence the symbiont genome sequence. And now our data set comprises more than 500 symbiont genomes. We now have many samples also from the deep sea, and this was very much thanks to collaborations with museums and the expedition in the Philippines that I was talking about before. Our collaborators have been very generous in sharing samples, and so those in the boxes here are just representatives of some of these deep sea lucinids that we have now in our data set. Okay, so just to walk you through this, most of the lucinids that we've sequenced so far, most of the symbionts we've sequenced so far, all belong to one family of sulfur oxidizing gamma proteobacteria. This is just not showing all of them here, but a representative set, and most of them are associate with Allochromatium venosum, a cultivated, very well-known sulfur oxidizing bacterium. There is a cultured relative of lucynid symbionts. We don't have any of them in pure culture yet, but there is a cultured relative called Sedimenticola, and it was cultured from salt marshes in the US. So they all fall into this one family. So this is really an ancient symbiosis that's diversified over many, many years. It's interesting that the closest symbiotic relative comes from the deep sea. You might know of the animal Riftia pachyptula. It's this enormous tube worm that's found at hydrothermal vents. It relies entirely on chemosynthetic symbionts for nutrition. And these chemosynthetic symbionts are actually closely related. They belong to the same family as the symbionts of lucynid clams. So there are links between deep sea and shallow water chemosynthetic symbioses that I maybe wasn't even aware of when I thought I was moving into a new field looking at shallow water symbioses, but there are very clear links between the deep sea and the shallow water. By now, we've identified many tens of different species level groups of symbionts. So although they all belong to the same family, they have also diversified into many different genotypes of symbionts. this diversity of symbionts can clusters into two different genera. So basically we have one family of chemosynthetic symbionts that associate with leucinoids, but they cluster into two distinct genera. One is called sedimenticola because it contains these cultured free-living sulfur oxidizers, and the other we called thiodeazotropha because this refers to its sulfur metabolism, thio, and deazotropha referred to its nitrogen fixing capability. However, this might have been a mistake in retrospect. The genus that we've called thiodeazotropha actually contains many members that do not seem to have the capability to fix nitrogen. So those shown in green are the ones that have this entire cluster of NIF genes in their genomes, and those shown in black are lacking this cluster. And so we do have quite a few members of the family that do not have the genomic capability to fix nitrogen. And one thing that all of these have in common, almost all of these have in common, is that these are these deep water samples. They all come from depths that are below about 30 meters in the oceans. So there's clearly some link between inhabiting the deep sea and not being able to fix nitrogen. So the question is, you know, there's losing metabolic capabilities is actually quite common in bacteria. There's this concept called use it or lose it, especially in symbiosis. If there's a function that you're not using, the bacteria will tend to lose it over evolution. And then the question is it been lost? Did they have it previously? Did ancestors have this capability and it was subsequently lost? Or did they just never have it in the first place? And those relatives that have this capability have acquired it, as bacteria can acquire metabolic capabilities through horizontal gene transfer. Clearly, there's a dynamic evolutionary history of nitrogen fixation, with some symbionts being able to, some not. Since the common ancestor, or the ancestors, Ilochromatium venosum can fix nitrogen, and the free-living sedimenticola sulfur oxidizers can fix nitrogen, we think that it was an ancestral trait of this family of sedimenticolaceae and that it has been lost multiple times. And this loss is often linked to environmental conditions. So colonization of the deep sea, that's typically not nitrogen limited. You might have seen some exceptions here, some that were not colored in blue. These are all symbionts of a species called Leucinoma borealis. And I'm mentioning this to you as well because it's also found in Sweden. So it colonizes the coastline of the United Kingdom and is also found in very shallow waters in Sweden. So the samples that we collected in Sweden were found from a depth of about 20 meters. So this is an exception. We've got a shallow water inhabiting clam. In the UK, it's even in the intertidal. So you could walk out into the sediments, into mud flats at low tide and just collect these bivalves. And this is really an obvious exception to this otherwise very clear pattern of shallow water species having nitrogen fixation, deep water species missing it. But Leucinoma is also an interesting exception for another reason. This is so just to show you a picture of Leucinoma. If you're ever on the coastline in Sweden and you see shells like this, I'd be excited to hear it. We went out to the Kristineberg marine station with a ship to sample these Leucinoma. This was a very interesting link. But Leucinoma is also interesting because it's one of the very few species that's found at high latitudes. The United Kingdom and Sweden is obviously, these are also typically, most of the samples we have, if you remember the map I was showing before, most are from tropical regions or subtropical regions. But the UK and Sweden are outside of these tropical regions, as I don't need to tell you. And these are also, if you look at models of nitrogen, marine nitrogen availability, which you may have done during your studies so far, these are also areas where nitrogen is quite available compared to the tropical and subtropical regions of the Earth. There's this sort of 45-degree magic cutoff. I'm sure Rachel can tell me the current status of these models, but typically at these latitudes, the nitrogen is more available than in lower latitudes. And so this would also match. So even though Leucinoma is a shallow water species, it's found at latitudes at which generally nitrogen is more available than in the lower latitudes. And that might explain why it's able to survive there without being able to fix nitrogen, or why this expensive metabolic process is not needed by the symbionts there. So just to sum up, through our work, we found that marine Leucinid clams associate with symbiotic all-rounders that fix sulfur, that oxidize sulfur, they fix carbon, and they can fix nitrogen. And this is a unique strategy compared to other well-known nitrogen-fixing symbioses, for example, in corals, where it's only one symbiont that's responsible for nitrogen provision, or symbionts that only provide nitrogen, or in plant nodules, where it's an exchange of carbon and nitrogen. We also showed that there may be a role for symbiotic nitrogen fixation in shallow water ecosystems that are inhabited by leucinids if they are releasing ammonium to the surrounding sediments. And our sort of large-scale evolution project has shown that the nitrogen fixing capability has probably been lost several times during evolution, and this is strongly colonized to the environment, to invasion of deeper waters by the animals and their symbionts. And this is an example of environmental selection for maintaining this expensive metabolism only in the shallow water habitats and mostly at low latitudes. So I would like to thank the team that I get to work with, even from afar, and the funding agencies, particularly the Vienna Science and Technology Fund, which was mentioned before. They helped me set up the lab and explore the world for leucinids, and a number of other funding agencies. Thanks for joining in and listening, and I'm sure you've got a lot of questions, so I'm looking forward to the discussion.